Immunohistochemistry, or IHC, offers spatial visualization and location of an antigen in tissue sections. This application can be challenging, with problems generally falling into two categories: weak or no stain and high background. Here are some troubleshooting tips to address high background for each of the 5 steps for successful IHC. Insufficient washing can lead to high background due to traces of fixative. Be sure to wash thoroughly and repeat as directed. Inadequate fixation can lead to diffusion of antigen in the tissue. Be sure to follow fixation directions precisely. Diffuse staining can be caused by tissue damage. Handle tissue carefully during fixation to preserve integrity. Insufficient penetration of detection reagents can be resolved by preparing thinner tissue sections. Different fixatives affect tissue antigen differently, so be sure to test and optimize the pH, incubation time, and temperature. Issues with the antigen retrieval step typically do not result in high background. However, it is important to use the proper antigen unmasking method, preferably via microwave, as prescribed by the fixation method. Possible reasons for high background that occur during the blocking step include interference from endogenous peroxidases or phosphatases, which can be resolved by quenching the enzymes with 3% hydrogen peroxide in methanol or water. A product such as Thermo Scientific's peroxidase suppressor can also be used. Alternatively, treat with enzyme inhibitors for alkaline phosphatases. Another reason for high background can be interference from endogenous biotin activity. Treat this by blocking with the avidin biotin blocking reagent before incubating primary antibodies. Lastly, interference from endogenous enzymes can also cause high background. Drain the excess buffer instead of washing the tissue sample prior to the addition of the antibodies. Possible reasons for high background at the detection step can include nonspecific binding of primary antibody. If the concentration is too high, it elicits high background and nonspecific binding. Try using a higher dilution of the primary antibody. There can also be nonspecific binding of secondary antibody. Treat with normal serum from the same species as the secondary antibody. If the reason is nonspecific binding, try using a higher dilution of the secondary antibody. Secondary antibody can cause nonspecific signal by reacting with tissue proteins. In this case, it is best to switch to a pre-adsorbed, affinity-purified secondary antibody such as those offered by Thermo Fisher Scientific. Sometimes high incubation temperatures can interfere with target detection. Solve this by lowering the temperature, even using a 4-degree incubation. Some formalin or paraformaldehyde fixatives cause autofluorescence in the green spectrum inflorescence IHC. Solve this by using a fluorophore in the red spectrum. If there is nonspecific binding of polyclonal antibody, try using a monoclonal primary antibody instead of a polyclonal. Lastly, there could be a substrate problem. Reducing the substrate incubation time, diluting the substrate, or changing to a different substrate source can address this. Once you perfected your staining, proceed to visualize your stained sample using the EVOS FL Auto 2 imaging system. Immunohistochemistry can be a challenging application. These troubleshooting tips can help you achieve the publication quality images you deserve. Find recommended products and protocols at thermofisher.com/IHC 5 steps.